last time about aspects of immune response kinetics, and we talked about sort of this idea of what's really happening throughout this primary response, the changes and maturation that the cells are undergoing, the difference between a naive cell and an effector cell, sort of the timing that we're seeing here. Remember, the effector cells were sort of short-lived, so that does limit the length of the primary response, but also the way that the quality of that response was changing over time, and how that can in fact impact what might happen if you have an infection that happens kind of while that primary response is still ongoing. I also mentioned that some of the cells that you have at the end of the primary response are a different type of cell called a memory cell. And one thing you should notice here is that we do have an increase in the quantity of cells specific for whatever our microbe is at the end of the primary response compared to at the beginning. So there is an increase in quantity, but there also is this massive change in quality where these cells have become memory cells. And today, we're really going to talk about that memory response and the specifics of those memory cells. And then we can also talk a little bit about some consequences of memory responses. So here you can see this general idea of what's going on with these both effector and memory cells. This figure from your textbook on the left is indicating the size of the CD4 and the CD8 T cell response over time relative to where they started. So you can see that massive increase. And you can see that we are going to be at a reduced level of those cells by a month or so after. That includes a lot of expansion of those effector cells, which will then, the majority of which will then die, and some fraction of those cells surviving to become memory cells. The memory cells do have some different properties compared to the effector cells. So the effector cells are going to be these short-lived cells that are really able to act dramatically against microbes, while the memory cells have this property of being long-lived. So they are going to live for a particularly long time to protect you later. So the ability to be long-lived is one of the special qualities. And memory B cells, in fact, are also, so B cells are also undergoing this process of differentiation into effector cells, specifically the plasma cells or memory cells. I am basically always today, or most of the time today, going to be using T cells as my example. Well, with having done a dissertation on memory T cell development, that's the place my brain goes as an example. I know I do have some B cell examples, but know that much of what we're talking about does apply to both T cells and B cells. In fact, if you want to read about B cell memory, you can read my sister's dissertation. So one of the really key things in thinking about memory cells, like I said, is lifespan. One of the qualitative differences about these cells is their ability to survive. And these data give you an idea of exactly what I mean by that lifespan change. So this, both of them are really actually getting at data from the same set of studies. In this set of studies, what was done was that people who received a smallpox vaccine were studied. The smallpox vaccine has not been given since the late 1970s. If you were born after the late 1970s, you have not received a smallpox vaccine, no matter where in the world you were born, unless you have done research on poxviruses. We've stopped giving that vaccine. So I am the only person in the room who's been vaccinated, and I've been vaccinated because of working on poxviruses, not because I was born early enough to get a smallpox vaccine. So people who have gotten a smallpox vaccine are relatively old at this point. And the virus was actually eradicated The last natural case that was seen was in 1977. The last case total was a little bit later than that, but the virus was actually officially declared no longer present on the Earth in 1979. So what we know is that there are all these people who were vaccinated, and we know when they were vaccinated. They have a little card, but then they never got infected. So we know what day they saw the antigens, and they've never seen them again. And so we can actually track how long did the memory cells live. We don't have to worry about did they get reinfected or what's going on. We can actually look how long they lasted. And so here in the left, you can see sort of this decline of the antibody response and the CD4 and CD8 memory response in years after vaccination. And so you can see that, yeah, the peak is, in 5 years or so. But you can also see that we can easily detect memory cells at 30 years post-vaccination. And in fact, if you actually look at some other data from the same set of studies, at the top you can see the antibody levels. The dotted line is where unvaccinated people are. And so you can see out to almost 80 years post-vaccine, we're detecting antibodies from that vaccine. So these memory cells, when I say living a long time, I mean a long time. Here you can see the same data for the CD4 and CD8 T cells. I did not remember to look up the difference between the colors of the bars in this, but I'm going to make an educated guess and tell you it's males and females. And what you can see is that even out in this 51 to 75 years post-vaccination, more than half of all of the individuals had CD4 and CD8 memory cells. So again, when I say these are long-lived cells, or long-lived responses, they are very long-lived. This doesn't necessarily mean the same memory cell lived for 70 years. It may have divided and had some progeny that were also long-lived. But one of the general key things here is that these responses are lasting for a very, very long time. And in fact, in this figure from your textbook, you can see part of the reason for that, which is that some proteins that are really important for survival, like BCL2, which stops apoptosis, are highly expressed in these cells. So they have a lot of survival proteins. And that is in fact one of the changes between a memory cell compared to, say, an effector cell or a naive cell. So this figure on the left is showing you levels of some different proteins in naive cells, effector cells, and memory cells. You can see that there are some proteins that get turned on when a cell basically sees antigen. So if the cell's naive, it doesn't really have much. As soon as the cell sees antigen, whether it's effector or memory, you've got more of that. Those are often thought of as the activation markers. So there are a bunch of activation markers that increase on those cells. There are some markers that go completely away when the cell is activated. But then there are others that we can see that and kind of think about them. So there are things like CD62L, L-selectin, as well as CCR7. We've seen both of those proteins this semester. Those are both proteins that we saw in trafficking, in terms of trafficking to a lymph node. A naive cell wants to go to a lymph node. It has a lot of those trafficking proteins because it wants to go to a lymph node in order to be able to find antigen. The effector cell does not want to be able to go to a lymph node, so it gets rid of those proteins that are needed for trafficking to a lymph node. Some types of memory cells will have those again, so the memory cells can go to the lymph node, be looking for antigen that might have come back, as well as get some sort of survival advantages from the lymph node. You can also see that there are some proteins that only effector cells make because of activation. Some special examples of that are things like granzyme B. The cell is really ready to be an effector. It's really ready to kill cell types or kill infected cells. So It's going to make a lot of these effector molecules. But there are also some proteins that are really specifically made by memory cells, like BCL2, I mentioned before, which is a really important cell survival protein to combat apoptosis, as well as a number of other proteins. And so we are able to look at T cells and distinguish different memory cells from them quite well, and distinguish different subtypes of memory cells. So if I get some T cells from a person, I can do some flow cytometry, and I can say these cells are naive, these cells are effector, these cells are memory. And when I do that, I even find that there are multiple types of memory cells. So the memory cells can be subdivided in a bunch of different types. Two of those are listed on the right here, which are the central memory and the effector memory cells, which differ based on things like which parts of the body do they traffic to, and how closely related to an effector cell are they? How quickly are they going to sort of trigger? There are also some other differences between naive and memory cells, and those are listed here. So this is a point that becomes really important in a few places, so that's why I kind of made little drawings, or found little drawings to show you this. In a naive cell, if we think about the genes that are required or that are used to transcribe cytokines, and really the genes that get transcribed whenever we are going to make some activation protein. So the genes encoding any protein needed for T cell activation in our naive T cell. Our T cell has never before seen antigen. The DNA in that area is called closed chromatin. You can see an example of closed chromatin here. The basic idea is that the DNA is wrapped really tightly around the histones. You can see that example here. If you wanted to use this DNA, if you wanted to transcribe something from this DNA, it would be hard. Because the DNA is really tightly wound. It's all packed closely together. The polymerase can't get in there very easily, nor can the transcription factors. And so if you want to transcribe a gene in those naive T cells, if you want to transcribe one of those the genes encoding a protein needed for activation, you need to open the chromatin. And the key thing that I want you to know, because some of you will have taken molecular genetics and thought about chromatin opening and closing, but some of you won't, is that's like another thing you gotta do. And the moral of the story here is that you have to signal that cell a lot in order to actually open the chromatin, get the transcription, do all the work. In fact, that cell, you can also see, doesn't really have much in the way of pre-made transcription factors. So it's got to make the transcription factors, got to open the chromatin, got to do a lot of work. You really have to smack that cell, give it a lot of signal in order to be able to transcribe all the things you need for activation. That cell is going to take a lot of work to activate it. For example, you might need a signal 1 and a signal 2 and a signal 3. With any of our cells that are activated, and this is listed here as memory cells, though in fact it's somewhat true of effector cells too, that same DNA, that same chromatin is much more open. And so you can see there are big pieces of DNA that are sort of free here. They're not all tied up around the histone. So the transcription is generally, the chromatin in this area for all of the activation related genes is open. It's not as hard to activate the cell. And in fact, a bunch of the transcription factors are already even made sitting around the cell ready to go. That cell is really, really ready to be activated. It doesn't, and so as a result, this cell is not going to take much in terms of getting activated. It does not require nearly as much signal in order to be activated. And so very importantly, and this is very important for both In fact, this is important for both memory and effector cells. Basically, once any of our T cells are affected, they do not need CD28 for co-stim. So once you've been activated, once you've grown up and are no longer a naive cell, once you've seen antigen one time in the lymph node from that dendritic cell, you don't need CD28 anymore. And that's part of the reason why it was so important to talk about dendritic cells when we talked about CD28 and co-stim. Dendritic cells are really good at making co-stim. I told you before, they were the best. And so a naive T cell really needs a dendritic cell to help it get turned on, because it needs a dendritic cell to give it all sorts of good helper proteins in order to turn it on, because it's so very needy. It needs a lot of attention to get turned on. When the cell's been activated, now it's going to act more easily. You can imagine if we have a naive T cell that is responding to a respiratory virus, like SARS-CoV-2, it's going to get activated in the lymph node by a dendritic cell that is presenting SARS-CoV-2 antigen. That is going to require signal 1 and signal 2. It's going to happen in the lymph node using a dendritic cell. We're going to make that T cell activated. Then that T cell is going to leave and go to the lung, where it might find some virus and be ready to kill the virus. It's now going to be seeing antigens presented by lung cells, not dendritic cells, because the lung cells are actually the ones activated. The lung cells might not be really good at giving B7. The lung cells aren't the all-stars that dendritic cells are in terms of T cell activation. But that's okay, because our cell that has been activated doesn't need that anymore. It is not as hard to activate that cell. And so this is listed as we don't need CD28 co-stim for memory cells. It's actually also true for effector cells. This will be an important point later on in a week or two when we talk about autoimmunity. These cells are going to be producing a lot more cytokine. They also sort of are going to be a bit needy in terms of getting some cytokine to help them live for their exceptionally long times, to keep them surviving. And as I mentioned before, there are many different types of memory cells. For a long time, we really thought a lot about two kinds, like central memory and effector memory. Central memory basically just went to lymph nodes to check if the infection was there. And they're in some ways what people think about as like the classic all-star memory cells. There are some effector memories that spend some time in lymphoid tissue, but also are patrolling the tissues a little bit. And we've been learning more and more that there are also some cells called resident memory cells that just live in tissues. So you might get some memory CD8s responsive to SARS-CoV-2 after you've been infected that live in the lung, because they think the lung is a good place to find my antigen. I should probably stay here. And so one of the places where immunologists have been learning a lot in more recent years is about these resident memory cells. Kind of hard, because if you wanted to study lung resident memory cells, you would have to get samples of T cells that live in your lung, which people are not going to just give you freely. Unlike blood, which is much easier to get, you can see central memory and effector memory spend some time in the blood. The resident memories don't. And so that's part of the reason why we're just now learning more about resident memory. And so we can see a whole lot of different types of memory cells. It's pretty easy to distinguish many of these cell types based on different flow cytometry markers. So there are lots and lots of different types of memory types of cells that people discuss in the literature. They are all distinguishable using different flow cytometry markers. And so this is just kind of showing you that there are these different subsets of memory cells. As a result of all of those differences that memory cells have compared to naive cells. The second time you are infected with a particular microbe, you get a response that looks like this, which is known as a secondary response. So you can see our primary response here. We are infected with our virus. It takes a little while for our adaptive immune response to start to come up. And during that time, the virus is sort of just battling with the innate immune system. You're going to start to feel sick. Eventually, the adaptive immune response peaks, and that allows you to clear that virus and get rid of the virus. And shortly thereafter, you start feeling better. One thing that this does not show on this slide is the amount of virus. It just says virus, kind of yes or no. So know that that is a weakness here. I actually made this slide in 2009. I have used this slide more times in the past year and a half when talking about some aspects of SARS-CoV-2 vaccines that I like, good job, 2009 self, made this slide, it's very useful. Because this shows you what can happen with a secondary immune response. And it shows a very key piece of information that I think is missed in some discussions of all of this. So this shows there's been a bit of time since we had our primary response. You will notice we did have a quantitative difference. We have more of our B cells, T cells, whatever cells you feel like thinking of when you look at this graph. We do have more after than we did before. But we have some period of time, and we might encounter that virus again in our life. And so here is our second encounter with that virus. These cells are memory cells. They're not naive cells. As a result of all of the changes that memory cells have, the fact that they are easier to stimulate, for example, and the fact that you have more of them, but mostly because they're easier to stimulate, you get this expansion of the response much faster. So you can see that we don't have that delay. In fact, here, the adaptive immune response and the innate response are happening basically at the same time, as opposed to in the primary where they're not happening at the same time. This response is actually faster, so that's a qualitative difference. It's bigger. These cells actually divide better, so you get a bigger increase than you did before. So that's sort of both a qualitative and a quantitative. And these cells actually function better too. And as a result, that virus gets stopped in its tracks. And it does not actually lead to further much replication. So what you can see here is the virus is only present for a very short amount of time. It's present for such a short amount of time you never feel sick. And this doesn't show you amount of virus, but if it did show you amount of virus, it would also go to a much smaller amount than it did the first time. And as a result, if I ever asked you, did you get infected with this virus a second time? You would say, oh no, absolutely not. I never felt sick. What are you talking about? I didn't get infected a second time. You really did, because infection, remember, just means a microbe getting into your body and colonizing you. It doesn't have anything to do with whether or not you feel sick. So you had such a small infection that you didn't feel sick. And if I asked you, you would not know. If I did very sensitive tests on you, I could find out that yes, you were infected a second time, but you would never know about that. This is why people talk about the idea that you can't get the chickenpox twice. It's tricky because A, you guys have never had a chickenpox. And B, this is also where now someone tells me about their cousin who got chickenpox twice. You in fact can get infected a second time, it's just that you never know because your body clears it so quickly. This is something that I think is really important, and the reason why I've been using this slide, and there's a similar one that I've made that we'll see later in the semester a lot, because people keep talking about breakthrough infections with SARS-CoV-2. Oh my gosh, you got your vaccine or you got infected and then you got infected again with a breakthrough infection. Yup, you did. You got this. And we used a super sensitive test to see if there was any virus there. You didn't feel sick. You wouldn't have known if we hadn't used that super sensitive test. We don't use that super sensitive test in any other situation. But we are using it right now, and so we're suddenly seeing this infection, whereas we never saw it before. But this is like not new. It's not scary. Not anything that's a surprise. You can see from my slide I made in 2009. There is an infection the second time. It's just that your immune system, one of my podcast colleagues talks about this as a fire extinguisher. Your immune system is such a good fire extinguisher, it puts it out before you actually know it's even there. So realize, again, I've used this slide so much now. Yeah, Jamie.
In the case of like immunocompromised people, is it then possible to have illness with a second infection?
It is. And realistically, I mean, it's theoretically possible for anybody. It's just much, much lower probability. But in particular, if one is immunocompromised and they don't make a good secondary response, then they're more likely to have some kind of illness. Absolutely. Yeah, Justin.
Say someone wants to be infected the third time, will the response either be greater or the same?
Great question. So in fact, if a person is infected a third time, they would experience something called a tertiary response, which would be even faster and even higher. And if they were infected a fourth time, they would have what's called a quaternary response, which would be even faster and even higher. So yeah, you do kind of, there is some sort of max level, but how you would get there, whether you would get there, is sort of theoretical and different from person to person. So imagine it that it gets better every single time. It just keeps on being faster and it keeps on being higher each time. So this is kind of the general idea of the difference between primary and secondary responses. Yep, Sebastian.
Is that kind of like how people, they become resistant to poison by taking it a bunch of times?
Sometimes. There's a lot more going on with poisons. And so there are a few different things that could be going on in a poison situation. In some cases, yes. In some cases, it is not immune related. Is the easiest answer to that. This table also sort of gets at some of those details about the differences between primary and secondary responses. You can see that at the primary response, we have a relatively small number of cells, whereas with the secondary, we've got an increased number. So yeah, there's a difference in number. But there's no delay in the primary response, or there's a delay in primary, there's no delay in secondary, because those cells are actually better. If we think about how the activation, it's really hard to activate the cells for a primary. It's very easy to activate for a secondary. You can see sort of the information about delay. You can see the information about innate immunity. So innate immunity is acting alone during the primary. The innate and adaptive are working together during the secondary. We can also see some information about antibodies here, so that we can think of some specific things about antibody responses. In particular, in a primary, you haven't had time for class switch. So at the start, you don't have IgG. You don't have somatic hypermutation. In a secondary response, you start with IgG, and you start with high affinity antibodies, because you've already gone through the processes we talked about in past lectures. You can see that from your textbook here as well. So you can see with our primary response, we've got this lag, and then we have an increase in antibodies. The black line is all antibodies together. But what you'll notice is at the beginning, all antibodies are IgM, which is the green. So basically, you're only making IgM. Eventually, you start making things other than IgM, particularly IgG. And so you start having IgG kind of late in the process. If you make a secondary response, however, you can see that you largely are getting IgG dominating your response. You do get a little IgM. Notice the IgM is actually less than you got the first time around. We'll get into why that is as we go forward, but I wanted to show you that it comes out even here, that the IgM is in fact reduced. And you're largely getting IgG, which has all sorts of nice good functions, and has isotype switched. So this is another way that you can see how secondary responses are so great. So this is showing you at times, and this is actually showing you primary, secondary, and the tertiary, like Justin was asking about. First, just which antibodies you see. So you can see at the beginning, you mostly have M. After secondary or tertiary, you mostly have G. And this is showing you the affinity, so how much are those antibodies binding. You can see at the beginning, we mostly have weak. M increases a little bit, but not too much, whereas G really increases, particularly with those second and third exposures. And so these are reasons, again, why that secondary response in general is going to be so much better than the primary response. This is sort of another kind of similar type of thing, just showing you the details with the antibodies. So we're getting information about frequency changes, isotypes change, affinity changes, amount of hypermutation changes. And this table actually, again, shows the same thing. The only reason I add this table that feels really repetitive at this point is because it has the last piece on it that the other tables have not, which is that you really only see this secondary response in terms of fabulous antibodies happening for protein antigens. So you can make a primary response to anything. You can only make a good secondary response with the large number of memory cells, those cells that are really well differentiated, that are making IgG, that are making high affinity antibodies. You can only do all of that with a protein antigen. And why can you only do that with a protein antigen? Erin?
Because T cells only recognize proteins.
Because T cells only recognize proteins, and you need T cell help in order to get B cells to this point. That's also why when someone is making antibodies for the antibody store, they have to think about this because they want to make these super fab antibodies that we're seeing here. You can further see this information in this figure from your book. And so this kind of just gives us a little bit of some data, some comparisons. If you remember, I told you previously about CD40 and CD40 ligand interactions. And I told you that if there are patients who are missing CD40 or CD40 ligand, they have a disease called hyper-IgM syndrome, where they don't ever class switch and they don't ever have somatic hypermutation. It's very sad. And when I mentioned that, I told you that there was a third gene that could also be mutated that would give the exact same phenotype, the exact same disease. The third gene is AID. I just hadn't told you about AID yet, so I couldn't say AID was the third one. Here, and these data are basically showing you what happens in mice that either have or do not have AID. And so we can look at their primary and secondary responses. And of course, we're specifically looking at antibody responses here. The graph on the left is showing you IgM. The graph on the right is showing you IgG. The red dots are mice with AID. The white dots are mice without AID. And you can see the times after immunization, first and second here. So what you can see is that if we just look at the mice who have AID, they make a little more IgM over time after immunization. And after the second immunization, again, they make a little more. If you look at the AID deficient mice, if anything, they maybe make a little more IgM, because they can't class switch, so they don't have any choices other than IgM. Part of the reason for that. And they particularly make more in the second immunization. Again, that's for the reason I'm going to tell you about. The IgM is actually actively suppressed in some of these later time points. And then if we look at the IgG responses, you can see only the mice after first immunization make a nice IgG. After in their secondary response, they make lovely IgG, and the AID mice don't ever make IgG. They can't because they don't have AID. So this kind of shows you the details of that response. In these data, as well as in some of the other data that some of the other figures that I've shown you, you might note that IgM being a little bit low in certain cases. So I think it was really nicely shown here, where you can see that the IgM starts to kind of go away once the IgG is present. I also saw it, oops, yeah, I guess I didn't. But particularly if you look at the secondary response, the IgM is actually lower than it was at the beginning. And we can think a little bit about this situation and what's going on here. What cells, if you really think about it, in this time where you are getting your second infection, what kind of cells would be making the IgM? Hello.
This is why they pay me the big
That is why they pay you the big bucks. What cells make the IgM in this situation, Sebastian? B cells. What kind of B cells?
Maybe plasma cells?
They're making IgM.
Oh, yeah.
Who's making IgM?
Not plasma cells.
Not plasma cells. Sebastian.
Is it naive?
Naive B cells. Cells that are making IgM or haven't class switched. They're probably naive cells, right? Make sense? You think about it, at this day, when you get this second infection, if you had a choice, would you prefer to have naive cells acting or memory cells acting? Jamie would prefer memory cells. Why would you prefer memory cells, Jamie?
Because we were talking before about how memory cells have a better and faster response.
Yeah, memory cells are awesome. So if you had to choose between the two, you would choose memory cells. Why might you have to choose? Why not just let the naive cells do their thing while the memory cells are also doing their thing? Yeah, Rishi.
No, because the naive cells won't they won't be able to tackle the thing better.
So they're not going to be very effective. So the naive cells are not going to be super effective. They're going to actually come up late, most likely. So they might even come up too late. There will be some naive cells, they will come up, but they'll come up late. Sometimes the virus is already gone. Sometimes you might not get them because the virus is already gone, and you didn't even have enough time to get them. But there's one other thing. Why might you have to make a choice? Yeah, Jamie.
Would it have to do with like just the energy your body is putting into one or the other activating? Like why would you activate the naive cells when the energy you're putting into that is going to be wasted?
Yeah, you're basically wasting ATP at that point if you give some ATP to the naive cells. If you let the naive cells start responding, then that's ATP that you don't care to spend. You would rather spend it for the memory cells, right? And if you think about things like the room in your lymph node, if there's going to be somebody proliferating and taking up room in your lymph node, you'd rather have the memory B cells proliferating, not the naive B cells. So this is really interesting because it turns out a secondary B cell response or a later B cell response actually has the ability to turn off the naive response. So once you're making good sort of activated memory type B cells, you actually stop making the naive cells, you turn them off. And it's for all the reasons we've just talked about. The memory cells are preferable in lots of ways, timing, effect, all of that. And you don't want to waste the ATP or the space in your lymph node or any of that kind of stuff. And so we have this ability to actually have only the memory cells actually responding in the secondary response, and not the naive cells. We actively inhibit those naive cells. You can see the example of how this works over here on the right. Here, in yellow, is a naive B cell. Orange is the activated memory B cell. This is a primary response. Here's we only have naive B cells in a primary response. So don't really need to show one there because we haven't made it yet. Here's a naive B cell. It binds to antigen. It gets some signal. It gets activated. Not a shocker. But when we get here to the secondary response, to later parts of the response, you can see that there are some antibodies present. There's some IgM and some IgG present, right? Here you pretty much had 0 of those antibodies, or very, very little of those antibodies. Here you've got a bunch, or more. Those antibodies will also be binding to antigen in this case. So here you didn't have any antibodies bound to antigen because you hadn't made them yet. Here you got some antibodies binding to antigen that were already leftovers from your previous response. Those antibodies will bind to the antigen, and those naive B cells all have an Fc receptor. And so if there is an antibody attached to that antigen, that Fc receptor gives a negative signal and turns the naive B cell off. So the naive B cells are actively turned off here so that we don't actually make another naive response. However, we do make this nice memory response. The memory cells are not inhibited. They would have some antibodies on them, but there's no inhibitory receptor. And so the memory B cells are allowed to respond. The naive B cells are not allowed to respond. You can also see this exact same thing here, where we've got the antigen that also has antibody on its surface that's binding to the Fc receptor, and that's leading to a negative signal that blocks B cell receptor signaling. Yep.
How does the initial response then not turn itself off if all the naive cells express that Fc receptor?
Because when the naive B cell gets to starts making antibody, it's no longer naive, and it will have turned off the Fc receptor.
Okay.
So when the cell stops being naive, it turns off the Fc receptor. Yep, Jamie.
So we were talking before about how it's mainly in the secondary responses a lot of IgG, but some IgM. What leads to there still being some IgM?
So you can have some activated cells that actually won't switch away from IgM, that will stick with making IgM. So you'll have some of that. You might have some limited number of naive cells that are acting, but it is not the response that you're used to. Does this make sense? You see why this is important? This is actually another set of slides I've been using a lot in the past couple of years. There are some COVID patients who are treated with monoclonal antibodies. They get antibodies in their body to stop to kill a virus. So basically, they're not doing well. We treat them with antibodies, and we treat them with a lot of antibodies in order to get rid of the virus. As in, there's one somewhat famous example where the person got 8 grams of IgG. And some people say, and so there's this question of, well, if you are that person, will you make your own immune response to SARS-CoV-2? Because you're infected, you get infected, you start making a response, and then you get 8 grams of IgG. What's going to happen in that case? Or what is the sort of theory based on this?
Your naive response would turn off.
Yeah, you're going to actually actively turn off your naive response. So you may not actually make a good response yourself if you've been treated with those antibodies. And if, say, you were treated with those antibodies, you might have to wait a while before you got a vaccine to make sure those antibodies are gone from your system. And so, again, this exact phenomenon I've been talking about a lot because I keep having to answer this question. Yeah, Jamie.
Can you just repeat, so in this figure, what is the gray antibody?
The gray antibody
Yeah.
is the pre-existing antibody. So it's
Okay.
it's this antibody that was bound to the antigen, that then the antigen binds to the B cell, and that antibody is engaging an Fc receptor. So again, you can see kind of some consequences of some of this information. We can also think about a couple of other things with memory cells. I have been careful about how I talked about some of this. And this textbook does actually do this correctly in being vague about it as well. Your textbook is a little bit more, they're not quite as vague as I would like them to be. Which is that there's actually this massive debate on one piece of this in the field, which is basically about where the heck do the memory cells come from? The question that people are debating in the field is, when your naive cells get turned on, do they make some progeny who are memory cells and some who are effector cells? Or and so you can see that arrows showing it going both ways? Or do they all become effector cells and just the ones who live become memory cells? So you can also see there's an arrow that way. And so I think that a lot of people have this idea that the effector cells become memory cells, and that's not actually totally clear. I would say the data, in fact, argue against that. And so, yeah, this is again showing, according to your textbook, they become effector cells, and then some of them become memory cells. I'm not actually sure that's really how it works. In fact, there is this paper, it's one of the more beautiful papers I've ever seen. It's all imaging, and they're so cool. All the papers in this group are so cool. It actually seems to indicate that when that naive cell divides, it actually sets up its mitosis in a way so that it can make an effector cell and a memory cell to feed both. And so it actually divides like this on this plane with the ability to go into both things. And in a lot of ways, people are sometimes thinking about calling memory cells like stem memory cells because they can become new effector or new memory cells. And so they actually have some stem cell-like properties. And so there is this idea that no, they could come from totally separate T cells. Some of them could become memory cells. Some of them could become effector cells. This is super debated. We currently think that this is actually related to the amount of signal that the cell gets. If the cell gets a little bit of signal, it becomes a memory cell that has sort of self-renewal properties and can keep making other cell types. And as it gets more and more and more signal, it becomes an effector cell. It has fewer and fewer and fewer choices. So at the beginning, it becomes a memory cell that could do anything. And as it gets more signal, it becomes an effector cell that only has a choice of dying. And so this is really kind of what we think is happening right now, is that in fact memory cells turn into effector and not the other way around. So that paper I mentioned that was so beautiful, it was in Science. I had the hard copy of the journal, with the magazine. If you flip to the article after that one, the article after that one suggested the other model. They were happening so much at the same time, and there's still a lot of debate on all of this. So there are lots of pieces to this. One thing that this figure and some previous figures have also implied is that this is super dependent on cytokines. And so those memory cells are going to require specific cytokines both to become good memory cells and to live for their long lifespans. And in particular, one of the cytokines that's really important is IL-7. IL-7 you saw in B cell and T cell development, and it helped developing B cells and T cells live. It turns out it also helps memory cells live. It's basically the live cytokine, the cytokine that helps cells survive. But there are lots of other cytokines that are sort of important throughout this process. All of the cytokines that are shown in this figure are cytokines that are in the same family, which is the common gamma chain family. In fact, the one family of cytokines that I introduced to you as a family. And just to give you an idea of how this cytokine stuff works, and I promise this is not totally meant to be self-serving. This is some data of how that looks. So in this experiment, I infected mice with adenovirus at 2 different times, day 0 and day 56, so 8 weeks. And I was measuring virus-specific CD8 T cells with my cytometer. And I was looking at wild types, and so you can first see the wild types are in purple. You can see a nice primary response, and then you can see the secondary response really nicely here. And then I'm also using mice that were deficient in a cytokine receptor gene. So this is the IL-21 receptor alpha gene. It's another member of that same family of cytokines. And I gave those mice the same infection. You can see they actually make an identical magnitude primary response, but the cells can't live, so they decline faster. And if I give them a second dose of the virus, they make a primary response again. They don't make a secondary response. And so if you keep infecting these mice, they only make primary responses. They don't ever go to a secondary. And so cytokines are super important in making any part of this happen. And so this is just an example of exactly what's going on with those cytokines. All right, so now we can talk about other aspects of immune response kinetics. So one thing I will tell you, again, this gets at some of the arguments people have been making about infection. And if you remember, I showed you last time, I'll even go back to the data just to be fully clear. So I showed you some data last time. These are the data. These data are showing you the decline in antibodies in people who are infected with SARS-CoV-2 over time, right? And so if you go to the doctor right now, you can have your antibodies measured. You can give me a number of how much antibody you have. It would actually look very similar to this kind of data. You can say, oh my goodness, the sky is falling. I've lost my antibodies. And I showed you last time that, well, the quality has changed, so it's actually okay. But one other thing I want to show you, these are the data on actually memory B cell responses. Doesn't go quite out to a year, but it could. If you look at those memory B cell responses, these are actually all the dots are different people. The line is sort of best fit. You can see the memory B cells ain't going anywhere. This actually specific is the answer to your question earlier. Okay, you walked away, that's fine. You're asking this exact question. The memory B cells are perfectly fine. And so even if the antibody levels are declining, as they should, because the effector cells should be declining, we seem to have memory cells that are persisting perfectly well over a long period of time. Unfortunately, you can't go to the doctor and get your memory cells measured. This takes some pretty complex immunology that's not done in doctor's offices. And so your antibodies are not actually a great indicator of how much immunity you have, because at least if you're me, I would way rather have some memory cells than just have an antibody sitting around, because memory cells do all those amazing things I showed you. And similarly, people have looked at different types of memory T cells over time. This shows you the different times on when this was done. And the biggest thing that I notice here is if I look at, say, these data on different types of T cells, the different types of T cells are still changing in percentages, even out to 6 months. So in fact, this response is changing in quality in terms of T cell memory as well. So we're getting evolution of the response on both the B cell and the T cell side. We actually are moving towards more and more of the lighter blue cells, which are central memory, which at least in my head are the best possible ones. So also super excited that I made this slide earlier this week, because I found out last night, I have a podcast this afternoon, and I also have to read a paper for the podcast, and it's always a struggle to find time to read paper. And the paper I have to talk about this afternoon is this. I already read it. I was so happy. All right, so that's that info. I already told you about the info about the boost timing. But I want to, I hope I have time for this. This is like there are so many parts that I have left that are some of my favorites. So I want to kind of start working through this one sort of example case. And we may not have time for the whole thing. We may have to do the rest of the example case next time. Okay? But I want us to imagine a couple of scenarios. So the first scenario that we are going to imagine is a scenario of my mom. So we're comparing and contrasting me and my mom. The y-axis here, I drew the y-axis too close to the thing to really be able to write a label. Can't really verbally tell you what the label is. The label is the amount of adaptive response. You can imagine this as T cells, you can imagine this as B cells, you can imagine it as whichever one you like best. And the x-axis is going to be time. Okay? So once upon a time, my mom got infected with a virus. I'm going to give you a virus as an example virus. We're going to not worry about some of the specific details of this virus and virus infection, even though there's a little part of my head that's like, but. We're just going to go with it, okay? Once upon a time, my mom got infected with measles, okay? So, she made a primary response to measles. There's my mom's primary response to measles. And as time went on, maybe at some point some other little kid in her neighborhood had measles. She was out playing with that little kid, right? So she got infected with measles again. She never knew it. So she made a secondary response to measles. Okay? Then, sometime later in her life, she was babysitting other kids. And imagine if she didn't know it, but she ended up babysitting a kid with measles when she was like a teenager. Tertiary response. Then, no, this is not true, but we're going to pretend. I come along. I get measles as a kid. Don't pretend I have. So my mom's taking care of me when I have measles. When I have measles. Right? And on and on and on. My little sister gets measles. My mom was also an elementary school teacher. So maybe she's in contact with lots of little kids who get measles. So you can imagine how high my mom's immune response to measles would be. I can't draw that high. Okay? Now let's imagine my mom now. She is retired. She's a little older. Immune responses do seem to decline a bit with age. And she has not been around a person with the measles in many years. Technically, what does happen in these sort of situations is you do have a decline in your immune response. Memory responses don't always stay at this high level forever. They do undergo attrition. And so she does have kind of this asymptotic decline. And maybe, when she gets to this low level, she would be susceptible again. When it declines that much, she's going to potentially get sick again because her cells have declined so much. Do you think that's likely to happen? Why not?
She'll be dead by then.
Because look how long that took to get to that very low level of measles, right? She's going to be protected for her whole life. Now we're going to have me as a quick example, and then we'll be done for today, and I'll give you the rest of my part of this example because there's a cool application of this that I don't have time for today. Okay, so imagine I got measles. It's actually not true. I got a measles vaccine. Measles vaccine actually does work here. Got my measles vaccine. Got my measles boost. I have never once in my life met a person with measles. Measles was eradicated from the United States in 2000. I came back, but we'll say never once in my life have I ever met someone with measles. what's been happening to me for the rest of my life since I got my measles booster at age 6? What is it, Rishi?
Going down.
Am I ever going to get to a point where I am susceptible to measles? Yeah, probably. Suddenly my immune response is going to go down. We are realizing in a lot of cases that immune responses we used to think lasted your whole life don't actually. You were just getting some additional boosters. And we now know that there are some groups of people who are a little bit susceptible to things that we didn't know. In a bunch of vaccine situations, we're hoping to now give another dose of vaccine later in life to pop those back up. We're going to start on Monday with this exact same example, but we're going to do the example with me and my mom and chickenpox. With the talking a little bit about chickenpox, how chickenpox and shingles work. And so we're going to talk about how basically the moral of that story is going to be that I am clearly, people in my age bracket are so getting shingles eventually. Like I am so going to have the shingles. That's basically what we're going to learn, but we'll go through this exact same thing with chickenpox.